Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. This week we continue our exploration into the systems of the cell with the endomembrane system. As always, at this moment we are popping up the terms and definitions slide so that you guys can pause and grab the vocabulary we will be using during this video, or alternatively you can pop on over to my Patreon link in the description below and grab my entire PowerPoint presentation as a PDF for 100% free, but patrons do get access to this video and all materials that go along with it a day early. Let's begin our exploration of the endomembrane system with the basics. So, eukaryotic cells have these membrane-bound organelles. That's one of the things that defines a eukaryotic cell. It's these membranes that make up the endomembrane system. So that includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, vesicles, vacuoles, and the plasma membrane that engulfs the cell. Now, the plasma membrane is not technically inside the cell, but it is connected to the endomembrane system, as we will see later on in this video. So, to put it simply, the endomembrane system has to do with all these little plasma membranes within the cell, endo. Now, all these membranes are either connected physically to one another, like the endoplasmic reticulum to the nuclear envelope, or by vesicles, which are these little membrane-bound sacs that carry out different functions that we will explore in this video. And the big deal about these little endomembranes is that they divide the cells into these functional components. So like the organs in you, which I'm assuming if you are human watching this video, you do in fact have organs. Those organs divide your body into the little functional parts that carry out specific tasks. This endomembrane system is what helps kind of compartmentalize all of the things that go on in the cell and the organelles and the little cellular sized organs, which organelle basically means little organ. So organelles are analogous to bigger organs in a human. So basically the places where these different things happen. The first thing that we're going to really explore in this endomembrane system today is the endoplasmic reticulum. Now, technically, I guess I could start at the nuclear envelope, but since we explored the nucleus last week, there's not much of a reason to. The nuclear envelope is the membrane that encloses the nucleus, and it happens to be connected to the endoplasmic reticulum. And what the endoplasmic reticulum is basically this network of sacs and tubules all enclosed in these membranes, separating them from the rest of the cytoplasm and cytosol and all the other fun things inside the cell. And as I mentioned before, they are attached to the outer part of the nuclear envelope. And now the endoplasmic reticulum is made up of two parts, the rough and the smooth endoplasmic reticulum. So let's explore a little bit about what those are, because they each have their own distinct functions. Let's begin with the smooth endoplasmic reticulum. Now, from this moment on, we're going to refer to the endoplasmic reticulum as ER. Not that I don't love using big words to make myself feel smarter, but for time's sake, it is more convenient to use the abbreviation. The smooth ER got its name because it lacks ribosomes on the outer surface of the membrane, so it appears smooth when looked at in the microscopic images we do have of endoplasmic reticulums in different cells. The smooth ER itself has a variety of functions depending on what type of cell it's in. The specific functions of the smooth ER are the synthesis of lipids, especially steroids like the sex hormones, testosterone and estrogen. So cells in the ovaries and testes tend to have quite a bit of smooth ER because they are producing those specific steroids, which if we think back a few videos prior when we talked about lipids, steroids are a type of lipid. But the smooth ER is also used in the processing of drugs, alcohol, and other toxins, so cells in the liver tend to have quite a bit of smooth ER as well. But the production and processing of different molecules is not the only thing the smooth ER is used in. If you look in your muscle cells, you'll see a lot of smooth ER because it's used to store calcium ions. And calcium ions are important when you look at different fibers contracting and different ion pumps, but we will get into that in later videos. The other part of the ER is the rough ER. Now, the rough ER gets its name because it looks rough in these microscopic images. And the thing that makes it look rough are all the ribosomes that are bound to the outer surface. And what the rough ER does is it actually makes membranes. But that's not all. It's also the place where the proteins that may be secreted from certain cells is made as well. And a little quick and dirty of how this is done is that the ribosomes that are studded to the surface of the rough ER use the messenger RNA to make that polypeptide chain, and then some sugars may be added and the molecule will be folded up to make that 3D protein shape. And this was explained a little more in detail a few videos back when we were exploring proteins. But once that protein is made, and if it does have sugars bound, a little fun tidbit here is it's called a glycoprotein because it has glyco sugars bound. But this protein, glycoprotein, whatever it happens to be, will then reach the end where it will be kind of wrapped up in a nice little transport vesicle that will literally bud off the end of the endoplasmic reticulum and carry that protein all the way over to the Golgi apparatus, which is what we're going to look at right now. The Golgi apparatus is essentially the cellular processing plant, and it's made of a bunch of stacks of just flattened sacs. And cells that secrete more proteins will have more stacks in their particular Golgi apparatus. But how does this whole thing actually work? Well, there is a receiving end and a shipping end, basically. The receiving end will accept these vesicles coming from the rough ER. That fused vesicle will then add its contents to the stack that it fused to. Those products will then get modified as they move between the stacks and end up on the shipping end. And that final product will be enclosed in another transport vesicle that will bud off the Golgi apparatus, and then it will be shipped out to the plasma membrane or occasionally a different part of that cell. Usually the plasma membrane to then be shipped out somewhere else outside of the cell. Moving on from here, we're going to talk a little bit about lysosomes and digestion. And no, it's not the digestion that goes on in your stomach and intestines. This is cellular digestion. And what a lysosome is essentially a little sack of digestive enzymes with a slightly more acidic environment than the rest of the inside of that cell. So it's the proper acidity for the enzymes to function, but it also keeps the rest of the cell protected from those digestive enzymes so that the cell itself does not get broken down from the inside out. So if you have like a little single cellular organism that needs to engulf food from its environment, a food vacuole will kind of bud off once the food is engulfed and move on into the cell itself. It then will kind of fuse to the lysosome, which you can see here in the diagrams I have on the slide, and then those digestive enzymes will break down that food and the nutrients will be released into the cytosol to then be used in different parts of the cell for whatever functions they need to be used for. Lysosomes also work to break down damaged organelles. If, say, in the example here in the diagram, a mitochondrion is damaged, it'll get surrounded by another vesicle or vacuole. A lysosome will fuse, break it down the same way it did with food, and then the materials can be repurposed. And then in your white blood cells, lysosomes are also used to break down harmful bacteria that are then engulfed by those cells and help your immune system function. So the main idea here is lysosomes are like these little pockets with an acidic environment with digestive enzymes to help break stuff up and then reuse that stuff in other places or protect an organism from harmful bacteria in its environment. If you noticed in the diagram on the last slide, the thing that the food was in was called a food vacuole and got a little confused. Well, good news for you, because now we're talking about vacuoles. And what vacuoles are essentially fairly large vesicles with a variety of functions depending on what kind of cell they're in and what kind of organism they happen to exist in. Specifically, they're notable in plant cells for their functioning in the growth of the cell and storage of different chemicals and whatnot. They can also have digestive functions, especially in plants and fungi, but they can also contain food when food has been engulfed, like we saw on the previous slide, or in certain single-cell organisms, they help expel excess water, especially if the organism is in a freshwater environment, because of how the water will flow from areas of higher to lower concentration. And the insides of cells have a lot of stuff in them that's not water. When you look at that compared to fresh water, the inside of the cell has a relatively low concentration of water. So the way that some organisms have evolved to deal with that is to have these contractile vacuoles that help them expel that water so the cell does not burst, which we have a fun little picture of one of those organisms, a paramecium, here on this slide. And then the other photograph we have here is pointing out that central vacuole in a plant cell, which helps with the storage. Certain species have chemicals that are either toxic or not pleasant to bite into to help protect them from herbivory. They also function in growth and will expand and contract depending on water availability. That's one of the easiest experiments you see in a lot of entry-level biology classrooms when you're first looking at plant cells, is putting them in a saltwater versus freshwater environment, because you can actually see the vacuole expanding and contracting. And that vacuole is the one that helps the little cells grow into their mature sizes as well throughout the life of that particular plant cell. So vacuoles are basically these big pockets that have many different functions. A lot of times it has to do with storage of water and other chemicals and waste, whatever needs to be stored. The last thing we're going to talk about today are peroxisomes. Now, they're technically not something that originates from the endomembrane system, and their relationship to other organelles is still a bit of a mystery to scientists, but this is probably one of the most logical places to talk about them because they are another thing that does have a membrane within the cell. So technically that could make them part of this system. They function in the breakdown of toxins. Specifically, they have a bunch of enzymes in them. The first sets of enzymes will transfer hydrogen from the compounds that are being broken down to oxygen to make hydrogen peroxide, which is toxic. Then they have this second set of enzymes that will come in and then convert that hydrogen peroxide into water, which is not toxic. So they do function in that breakdown of toxins that other parts of the endomembrane system also functions in. And there are some peroxisomes that will break down fatty acids for use as cellular fuel, but again, their specific relationship to other organelles is still a bit of a mystery because science is frustrating and we still have more questions than answers, but that's also what makes it fun. So that's all I really have for you today about the endomembrane system, and I do hope that you got something out of this, and have a wonderful rest of your morning, day, evening, night, what have you. I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.